Last class period, we spoke about intercellular communication in general, and we learned a little bit about receptor tyrosine kinases. You just watched a video about how activation of a receptor tyrosine kinase can result in the activation of a monomeric G protein called RAS and the downstream effects of that RAS activation. I want to review that, but also point out that RAS is not the only second messenger that can become activated in response to receptor tyrosine kinase ligand binding. After reviewing the RAS pathway, I'll introduce a second pathway that is activated by receptor tyrosine kinase ligand binding. Epidermal growth factor, or EGF, is one example of a ligand whose binding to a receptor tyrosine kinase results in the activation of RAS. As you saw in the previous video, so I'll say this quickly, EGF is a dimeric growth factor that, when it binds to a receptor tyrosine kinase, results in the dimerization of these single-pass transmembrane receptors. Dimerization allows for cross-phosphorylation, meaning that the receptor on the right phosphorylates that on the left, and the receptor on the left phosphorylates the one on the right. Phosphorylation occurs on tyrosine amino acids of these receptors, and those phosphotyrosines are recognized by an adapter protein known as GRAB2. The previous video referred to this as GRB2. GRAB2 contains an SH2 domain that recognizes the phosphorylated tyrosines. Upon binding to the phosphotyrosines, GRAB2 changes shape, exposing a polyproline sequence that is recognized by the SH3 domain of a guanine nucleotide exchange factor known as son of 7-less. Son of 7-less, upon binding to GRAB2, becomes activated such that it can cause the release of GDP and the binding of GTP to RAS. GTP binding to RAS activates the protein such that the signal can be transmitted within the cell. One of the targets of activated RAS is MAP kinase. The previous video referred to this as RAF. MAP kinase, or RAF, is a protein kinase, so its downstream target is MAP kinase. The previous video referred to this as MEK. MEK is a kinase, so it will phosphorylate downstream targets, one of which is MAP kinase. The previous video referred to this as ERK. MAP kinase, or ERK, is, as the name implies, another MAP kinase, or another kinase. So it will phosphorylate downstream targets as indicated in this image, and each of these phosphorylated downstream targets becomes activated such that things can occur. In the case of EGF binding to its receptor tyrosine kinase, cell division is initiated. In addition to activating RAP kinase, another possibility exists. Other growth factors activating receptor tyrosine kinases can result in the downstream activation of alternative second messengers. One example is fibroblast growth factor, or FGF. When FGF binds to its receptor tyrosine kinase, phospholipids, specifically phosphatidylinositol, in the cell membrane become phosphorylated. This is because FGF binding to its receptor tyrosine kinase results in activation of an enzyme known as phosphatidylinositol kinase. We abbreviate this PI3K or PI3 kinase. PI3 kinase phosphorylates the polar head of phosphatidylinositol in 3 separate places, resulting in what we refer to as PIP3, phosphatidylinositol phosphorylated 3 times. In the absence of FGF binding, phosphatidylinositol is in the plasma membrane. It's part of the plasma membrane, and it's not recognized by the intracellular messengers that ultimately will be able to bind. In the presence of a signal molecule, specifically FGF in this case, the receptor tyrosine kinase becomes activated. It, in turn, activates PI3 kinase. PI3 kinase phosphorylates phosphatidylinositol, and 2 cytoplasmic proteins, second messengers, are able to bind to the phosphorylated phosphatidylinositol using their plextrin homology domains. These, in turn, become activated to activate downstream signals and elicit a cellular response. Looking at the specifics of fibroblast growth factor binding to its receptor tyrosine kinase, we see that FGF is a dimeric growth factor that, upon binding to its receptor tyrosine kinase, causes another receptor to come in close proximity. This dimerization of receptor tyrosine kinases that's brought about by binding to a dimeric ligand results in cross-phosphorylation on tyrosine amino acids. Phosphorylated tyrosine amino acids are recognized by the SH2 domain of PI3 kinase. PI3 kinase binding to phosphotyrosines on the receptor tyrosine kinase results in its activation such that it's able to phosphorylate phosphatidylinositol in the membrane. Phosphorylated phosphatidylinositol, or PIP3, then serves as a docking site for a couple of second messengers. These second messengers recognize phosphorylated phosphatidylinositol via their plextrin homology domains. This binding between the plextrin homology domains of these second messengers and phosphorylated phosphatidylinositol results in the tethering, if you will, of these second messengers to the plasma membrane. This brings both of these second messengers to the same place within the cell such that they can find each other at the cell membrane. One of these plextrin homology containing second messengers is the protein kinase that's shown in brown. This protein kinase's target is a second protein kinase known as AKT. AKT is phosphorylated by the brown protein kinase 1 at the membrane, and it's also phosphorylated by a second cytoplasmic protein kinase. Doubly phosphorylated AKT then releases its binding from PIP3 such that it's released into the cytoplasm so that it can phosphorylate downstream targets. AKT then is a protein kinase whose activity is activated when it's doubly phosphorylated and when it's not bound to PIP3. How does binding of FGF, or this survival factor, result in a cell staying alive? Recall that in the absence of survival factors, a cell will enter apoptosis, the process of apoptosis, and ultimately die. However, in the presence of survival factors, or FGF in this case, a cell will live. This occurs via the activity of AKT. FGF will bind to its receptor tyrosine kinase, and that results in activation of PI3 kinase. Activation of PI3 kinase results in phosphorylation of phosphatidylinositol that is recognized by the plextrin homology domain in AKT. AKT is phosphorylated by a couple of protein kinases, and therefore it's released from its association with PIP3 into the cytoplasm where it's active. One of its targets is a protein known as BAD. Active BAD functions to sequester a protein known as BCL2, keeping it inactive. When BCL2 is inactive, cells enter the process of apoptosis. However, in the presence of FGF and active AKT, AKT phosphorylates BAD, making it inactive. In the phosphorylated state, BAD is inactive because it's unable to bind to BCL2. BCL2, now liberated into the cytoplasm, free from BAD, is active. Active BCL2 is able to promote cell survival by inhibiting the process of apoptosis. This is one of the two targets of active AKT, and this is the mechanism by which cells survive in the presence of FGF. Another target of active AKT is a protein known as TOR. When TOR becomes phosphorylated by AKT, AKT being activated by FGF, alternative downstream targets result in cell growth or cell division. So FGF binding to its receptor tyrosine kinase can promote either cell survival through the inactivation of BAD, as shown in the previous video, or it can produce cell division via the activation of TOR.